In modern biology, we're just drowning in data. But what if the most important stories, the biggest breakthroughs, are getting lost in all that noise? Today, we're going to dig into a really powerful new method that scientists are using to find clear, simple narratives hidden inside overwhelmingly complex information. Okay, so let's just dive right into this with a question that scientists face every single day. When you're looking at thousands, maybe millions of data points, how do you find the actual narrative? How do you separate the signal from the static? That is the core problem we are tackling today. And the challenge really kicks off with the sheer scale of the information we can now collect. We are absolutely living in an era of biological data overload. To really get this, let's look at a field called metabolomics. Think of it like a chemical snapshot of what's happening inside a living thing at any given moment. So, for example, by studying these tiny molecules, we can see exactly what a yeast cell is doing during fermentation. It's an incredibly powerful technique, but man, it generates a ton of data. This slide just lays out this central dilemma perfectly. In the study we're looking at, scientists took just 53 samples, but for each and every one of those samples, they were tracking 114 different metabolites. This is what's known as the N is much less than P problem, where you have way more variables to measure than actual observations. It's kind of like trying to solve a huge mystery when you've got way more suspects than you have clues. So, with this absolute flood of data, how in the world do scientists even begin to make sense of it all? Well, they have a standard toolkit that they usually reach for to try and simplify things. The go-to method is called principal component analysis, or PCA for short. And the best way to think about it is like this. Imagine you have a really complex 3D sculpture. To understand its basic shape, you could just shine a light on it and look at its 2D shadow. PCA does something very similar with data. It takes this high-dimensional, super complex data set and projects it into a simpler, lower-dimensional view so we can actually spot the patterns. But of course, there's a catch. Just like with that shadow, you might miss some really crucial details of the original sculpture. The source paper for this explainer puts it perfectly. When you reduce all that complexity, useful information may be lost. Really important parts of the story can just disappear. And one of the most important pieces of information that often gets lost in the shuffle is the element of time. And this is the key weakness that the researchers really wanted to fix. So here's the crucial point. A standard PCA analysis treats all the data points as if they happened at the same time. It has no regard for their order. It's like taking the pages of a story, shuffling them all up, and then trying to figure out the plot. For a biological process like fermentation, which unfolds over hours, the sequence isn't just a minor detail, it is the entire story. So, to solve this problem, the researchers developed a much smarter way to look at the data, a new kind of lens that actually respects the arrow of time. They call it a differential penalty, which sounds kind of complicated, but the idea behind it is actually pretty simple. First, you look at the data in its proper time sequence. Then, the algorithm applies a penalty, like a little fine, anytime it sees a huge, jarring leap from one point in time to the next. And finally, it rewards and highlights the patterns that show a smooth, logical progression over time. It's designed to find the connected path, not just a random cloud of dots. Okay, so that's the theory. Sounds good, right? But does it actually work in the real world? Well, to find out, the researchers put it to the test on a very real-world problem. Their test case was all about biofuel production. They took 3 different strains of yeast and tracked those 114 metabolites over a 96-hour fermentation process. The whole goal was to understand the metabolic differences between the strains to hopefully make the ethanol production process a lot more efficient. And check this out. The results from the standard PCA method perfectly illustrate the problem. The analysis just gave them what the researchers called a messy, jumbled cloud of data. The 3 different yeast strains were all mixed up, and you had no clear sense of progression from hour 0 to hour 96. As the slide says, the story was completely lost in the noise. But when they applied their new smoothed PCA method, the result was a total night and day difference. The analysis revealed these 3 distinct, clean paths. Each one clearly traces the metabolic journey of a specific yeast strain over the entire 96-hour period. The story that was buried deep in the data suddenly became perfectly clear and so easy to read. So what does this all mean? Well, this new technique is so much more than just a way to make prettier graphs. It really represents a genuine step forward in how we do science. The big takeaway here is that by preserving that timeline in the data, scientists can get a much deeper understanding of these really complex processes. Seeing that clear path lets them pinpoint key metabolic changes, which can then be used to engineer better yeast strains and produce biofuels more efficiently. It's simple. Better tools lead to better insights, which in turn leads to better technology. And what's really cool is the paper shows there isn't just one magic bullet. The researchers developed a whole suite of tools for different jobs. Smoothed PCA, for instance, is fantastic for visualizing the process over time. But another method they developed, smoothed RFDA, is way better at clearly separating the different groups, in this case, telling those 3 yeast strains apart. So now, scientists can pick the right tool for the right question. And that really brings us to our final thought. This was a story about yeast and biofuel, sure, but the principle is universal. It just makes you wonder, in all the massive data sets being collected right now, in medicine, in climate science, everywhere, what other crucial stories are just sitting there, hidden in the noise, waiting for someone to apply a smoother lens?